My name is Mushtaq Ahmed, and I'm here today with Ahad Molvi. We are third-year medical students, and we'll be talking about tubular reabsorption and secretion. So I'll begin first. Regarding tubular reabsorption, we'll only discuss the high yield stuff that you'll be most likely asked in your vivas and in your BCQs. So regarding tubular reabsorption, you should know, first of all, that it is an active process as well as a passive process. And this is in contrast to filtration, which occurs across the Bowman's capsule from the glomerulus, which is only a passive process. You should know that tubular reabsorption is a highly selective process, and what it means is that you're returning important substances from the filtrate back into the blood. And this can occur via two mechanisms. You have paracellular reabsorption and you have transcellular reabsorption. Paracellular reabsorption, as the name implies, you're reabsorbing substances between the cells. So important examples that undergo paracellular reabsorption are calcium ions and magnesium ions. We'll be discussing this later on as well. Transcellular reabsorption, this is the process where you're reabsorbing substances across the cell membranes of the cell. So examples of transcellular reabsorption substances that undergo this are sodium ions, you have glucose, you have amino acids. And other types of transport mechanisms that are involved are simple diffusion, you have facilitated diffusion. An important example of simple diffusion is urea, primary active transport, secondary active transport, and pinocytosis. So what are some of the important transport proteins that undergo primary active transport? So you should know that sodium, potassium, ATPase pump, the hydrogen, potassium, ATPase pump, these are important transport proteins that are involved in primary active transport. And some important proteins that are involved in secondary active transport are the HGLT1 and HGLT2 transporters. You have the sodium hydrogen exchanger, you have the sodium chloride, the sodium potassium 2 chloride co-transporter, and pinocytosis. What pinocytosis essentially is this is the opposite of endocytosis in a sense. Endocytosis is, in simple words, cell eating. Pinocytosis is cell drinking. And this is the active process. It requires energy in the form of ATP. And this is how proteins that are filtered into the tubules are reabsorbed back into the PCT cells and eventually into the bloodstream. So pinocytosis is important regarding reabsorption of proteins. Now, regarding the proximal convoluted tubule, you should understand how the cells that are within the proximal convoluted tubule, how are they modified? How are they able to reabsorb substances? And these factors include that the PCT cells are rich in mitochondria, and that allows them to synthesize energy in the form of ATP, which they eventually use to reabsorb certain substances via active transport or via pinocytosis. In addition, you should know that the brush borders lining the luminal side of the PCT cells, the luminal side meaning the side of the cell that's facing the lumen, and it has a large surface area which helps in having large number of transport proteins and pumps to reabsorb the substances from the filtrate into the PCT cell and eventually into the bloodstream. It's very important to know, especially for your BCQs and Vivas sake, that the reabsorption that occurs in the PCT is isoosmotic. So it has the same osmolarity as that of the plasma or as that of the blood, which is 300 milliosmoles. So that's very important to know that the proximal convoluted tubule undergoes isoosmotic reabsorption. 65% of sodium and water are reabsorbed. And as I mentioned before, proteins are reabsorbed via pinocytosis. In addition, secretion also occur within the proximal convoluted tubule, and specifically secretion of drugs, organic acids, bases, and toxins. Now, we'll start with the reabsorption of glucose. We'll be discussing other substances, other ions as well. So regarding glucose, you should know that normally in a healthy, normal individual, no glucose should be present within the urine. All of it, 100% of it, should be reabsorbed back into the blood. However, if a patient, for example, with diabetes mellitus, where they have high elevated blood glucose concentrations, in such people, you'll find that glucose is present within the urine. Hence, it's called diabetes mellitus, sweet tasting urine. The renal threshold for glucose is 200 milligrams per deciliters. And this is very important to know for Viva as well as BCQs. And what renal threshold actually means is that For any substance, it is the plasma concentration at which a substance first appears in the urine. So regarding the renal threshold for glucose, this is the plasma concentration of glucose beyond which you will begin to see glucose appearing in the urine. So, and this value, according to Guyton, is 200 milligrams per deciliter. And beyond this, glucose will begin to appear in the urine. What are the important transport proteins that are involved for reabsorption of glucose? You have the sodium glucose co-transporters, and these are present in the early or the proximal part of the proximal convoluted tubule. In short, they're called SGLT1 and SGLT2. Now we'll move on to discussing how sodium, potassium, and phosphate are reabsorbed. In the proximal convoluted tubule, 65% of sodium and potassium are reabsorbed. And you have to understand how sodium is reabsorbed in the early PCT and in the late PCT. So in the early PCT, sodium is mainly reabsorbed via co-transport and co-transport with other substances like glucose, amino acids, phosphate, lactate. Sodium is also reabsorbed with via the sodium hydrogen exchanger, which transports sodium ions into the PCT cell, and in exchange, hydrogen ions leave the PCT cell and enter into the lumen. So in the early PCT cell, the most predominant way sodium is reabsorbed is via co-transport. On the other hand, in the late PCT, sodium is mainly reabsorbed along with chloride ions. So this is just an important differentiating point you have to know of regarding sodium's reabsorption. Phosphate is reabsorbed by the sodium phosphate co-transporter protein, and a very important hormone called parathyroid hormone. What it does is it inhibits this co-transport protein, the sodium phosphate co-transport. And what happens is phosphate is not reabsorbed, and as a result, phosphate is excreted out of our body. So parathyroid hormone, one of the effects of parathyroid hormone of PTH is that it increases excretion or removal of phosphate ions. Now, regarding resorption of bicarbonate ions, this is a little complex cycle that you should understand, especially for SPE. They can give you a diagram of this, and you should identify where's what. So you should know that resorption of bicarb ions occurs mainly in the early proximal convoluted tubule. And the cycle begins, so you should start, in order to make it easier, you should start inside the PCT cell. Inside the PCT cell, you have carbon dioxide and water molecules, which are combined together to form carbonic acid. And this is done with the help of the enzyme intracellular carbonic anhydrase. All right, this is intracellular because we are talking about inside the PCT cell. Once the carbonic acid is formed, it's going to dissociate into hydrogen ions and bicarbonate ions. Now, this bicarbonate ion is going to eventually leave the PCT cell and enter the bloodstream. It's going to be reabsorbed. However, the hydrogen ions, the H+ ions, on the other hand, they're going to leave the PCT cell with the help of the sodium hydrogen ion exchanger, and it's going to enter into the lumen of the proximal convoluted tubule. And once it enters the lumen, the H+ ions will meet the bicarbonate ions that were filtered, and they're going to combine together to form carbonic acid. And then once carbonic acid is formed, it's going to be broken down to form water and carbon dioxide with the help of the brush border carbonic anhydrase enzyme. And this carbon dioxide will eventually enter into the PCT cell, and this is how the cycle repeats. So the important thing is that the net result of this entire process is the reabsorption of bicarbonate ions. Next, we'll talk about resorption of other substances like amino acids, magnesium, calcium, and urea. You should know that 100% of all amino acids are reabsorbed in the proximal convolute tubule. And just like similarly, we mentioned glucose and amino acids, 100% of them should be reabsorbed in the PCT. Magnesium and calcium, as I mentioned before as well, they reabsorb via paracellular reabsorption, which is reabsorption between the cells. And about 50% of urea molecules are reabsorbed via simple diffusion in the PCT. Now, regarding renal plasma flow and clearance, there are a lot of formulas regarding clearance, regarding renal plasma flow, and other things as well, but they're not that high yield for your med school exams, and especially even for Viva, no one is going to ask you. What you should know, however, especially for BCQs, is that para-amino hippuric acid, PAH, is a marker for renal plasma flow. This is a very important question that they'll most likely ask you. And another important thing is that inulin is a marker for glomerular filtration rate, GFR. So these two important points, keep in mind as they can ask you this within your exams. Another thing which you should know is that alkaline urine causes increased excretion of acids from the body, and vice versa. If you have acidic urine, it'll cause decreased excretion of acids from the body. And if you have acidic urine, that's going to cause increased excretion of bases from the body, and vice versa. If you have alkaline urine, that's going to cause decreased excretion of bases from the body. Okay.